All right, so we left off on Wednesday and we talked about this idea that for a lot of microbes, oxygen is something that they have to be considerate of because there are a number of different microorganisms that have differing oxygen requirements. So we have some organisms that absolutely require oxygen, all the way to organisms that will be killed by oxygen. And so the reason that oxygen is toxic is because of the production of what we call reactive oxygen species. So by doing normal cellular metabolism, when exposed to oxygen, cells will produce these free radicals or reactive oxygen species. And those can damage DNA and proteins. And so the reason that microorganisms can't tolerate these reactive oxygen species is because many of them that live in these anaerobic conditions don't have enzymes to detoxify the reactive oxygen species. But things like us and other microorganisms that can deal with oxygen have enzymes that allow them to detoxify various reactive oxygen species. So we have catalase, which allows them to detoxify hydrogen peroxide. Peroxidase also allows the detoxification of hydrogen peroxide. Superoxide dismutase allows them to get rid of superoxide radicals, just like superoxide reductase does the same thing. So not only do microorganisms that live in aerobic environments have these enzymes, so do we. And in fact, one of our major defense strategies in the immune system is we produce in our immune system reactive oxygen species to kill microorganisms. And so these enzymes can allow microorganisms to grow in aerobic environments, but having these enzymes can also allow these microorganisms to fight our immune system. So the ability to make these enzymes can be a virulence factor because it allows them to continue to cause infection, but it also can allow them to live in an aerobic environment. And so when we test for catalase production, you'll do this when you do your biochemical testing towards the end of the semester. You just drop hydrogen peroxide on a colony of bacteria or on some bacteria that are smeared on a plate. If they produce bubbles, they produce catalase. If there are no bubbles, there is no catalase. Okay. So we talked a lot about different environments where microorganisms can find themselves: high heat, low temperature, low pH, high pH, oxygen, no oxygen. So now the last little part of this chapter talks about what do we do when we no longer want microorganisms to be somewhere. And this is something that as humans, we are very, very interested in. How do we get microorganisms to not be places we do not want them to be? And so we can control microbial growth. And so we have devised a number of different mechanisms that we can use to get rid of microbes. There are a number of different terms that we'll use. Sometimes in the general public, people will use these terms interchangeably, but they do not mean the same thing. So as a microbiology student, it's important that you understand the distinction between some of these terms. And so the first term that they give us here is decontamination. So decontamination is just the treatment of an object to make it safe to handle. So something that is decontaminated is not something that is sterile. It is not absent of life. It is simply safe to handle. Along similar lines, disinfection removes pathogens, hopefully, but not necessarily all microorganisms. And so for the most part, when you're talking about surfaces in your home, going out to eat and using utensils and glassware, the human body, you are talking about disinfection or decontamination. You are not talking about sterilization. However, there are some circumstances where having something be sterile is absolutely critical. And so there are mechanisms that we have designed in order to completely sterilize things. Most often we use heat to sterilize anything that we want to have be sterilized. And in general, we actually use moist heat instead of dry heat. So we usually use steam under pressure. And part of the reason that we have to do that is because of endospores. So remember, we talked about endospores very briefly. Do you guys remember what type of bacteria can form endospores? Gram-positive, especially bacillus. That's okay. Does start with the C. Anybody remember the other genus? Tetanus, botulism, Clostridium. Right? So Clostridium and Bacillus are the two major genera that can cause human disease that form endospores. And endospores are generally resistant to normal heat. So what we have to do is use heat, especially moist heat, so steam, under high pressure, and that actually will be enough to kill the endospores. And we do that in a device called an autoclave. So the autoclave that we have for our building, where we do microbiology labs, is just directly across the hall from microbiology lab. And so it's a sealed device that allows the temperature to get above 100 degrees. It actually gets to 121. And it has high pressure. And so that high pressure with that steam at 121 is enough to kill endospores. And so what we do as microbiologists who are dependent on autoclaves is we actually will put spores in the autoclave. We will run the autoclave and see if we can regrow the spores. If we can regrow the spores, that's a problem, and we need to get our autoclave fixed. So we test it with spores to make sure that the autoclave is working. But it's not the pressure that kills the organisms, it's the high temperature. But having that pressure allows the temperature to get even higher. So that's when we want to sterilize things. And the growth media, for the most part, that you use in the lab is sterilized in there. All of your waste that you put in the biohazard bin will go into the autoclave and be sterilized through there. If you ever have been to a tattoo parlor or piercing studio, they also usually have their own autoclaves that can fit on a bench top to sterilize the needles. If they don't go there. Most nail salons should have autoclaves too to sterilize their equipment. If they don't go there. Dentist's office also usually have autoclaves to sterilize their equipment. If they don't go there. So yeah, if you've ever gotten a tattoo or a piercing, they should open the little packet in front of you. Like, oh, cool. Yeah. If they don't open the packet in front of you, run as fast as you can. I don't care if you paid your deposit, you're going to get something gross. So autoclave, very, very important. But I'm talking about a lot of solid things like metal tools. Okay. But your liquid media, a lot of it can also be sterilized that way as well. But unfortunately, sometimes getting things up to that really high temperature can change, especially with liquids, can change the properties of the liquid. And if it's something that you want to drink, you often are concerned with the flavor. For example, any of you who drink milk, I am most certain that you would never drink milk that had been through the autoclave. It would be absolutely disgusting. Just picture if you put milk on the stove for like an hour. Boiling milk for an hour? Yeah. You want to drink that? No. So what we do instead is a process called pasteurization. So milk, orange juice, so things that you're going to consume are not sterile. So they use heat for a very, very specific period of time to a very, very specific temperature in order to reduce the microbial load as much as possible. But it does not kill all organisms. So if you buy a gallon of milk at the grocery store, even if you don't open it, and you just left it in your refrigerator the entire time, it will still go bad. There are still microorganisms in there. There is a USDA-approved level of microorganisms in your milk. There's also an USDA-approved level of fecal bacteria in your milk. Enjoy. Okay? So it doesn't kill microorganisms. It reduces the load. We can also use ultraviolet light to sterilize surfaces, because what ultraviolet light does is it causes breaks in DNA. Right? This is the same reason why they say if you go outside without sunscreen, you'll eventually get skin cancer, because UV light causes damage to your DNA, which can, in humans, cause cancer. In microorganisms, it usually kills them. So we use UV light in my lab in our biosafety level 2 hood. So we'll have a picture of that on this slide here. So when you close the sash, the UV light comes on, and every surface that is touched by the UV light should be sterilized by that light. But it cannot penetrate solid, opaque, or light-absorbing surfaces. So every time I go to a lab and they have their hood just full of garbage, I'm like, well, that's not doing you any good, because anywhere the light does not touch, the microbes are growing. It's like the Lion King, right? We don't go over there because the light doesn't touch it. Don't go over there. The light doesn't touch it, your microbes are growing. So UV light is really good for sterilizing a surface like this. We can also use other types of radiation, not just UV radiation, but we can use ionizing radiation. There are some microorganisms, though, that are still quite resistant to radiation. So it's not going to get rid of everything, but we often use radiation to sterilize as much as possible things that we can't autoclave, like plastics. So if you ever find yourself working in a lab for any sort of extended period of time and somebody runs plastic through the autoclave, it's a very unfortunate experience because plastic melts. So lots of surgical tools, like scopes that are made of plastic. You can use radiation to sterilize them. Okay, a lot of drugs, so like antibiotics, any sort of pharmaceuticals, because if you heat them up, it will denature them and they won't work properly. So the best way to sterilize them is through radiation. We also irradiate a lot of our produce and meat to get microbes off the surface to, again, try to extend the shelf life as much as possible. And it actually works pretty well. You can extend the shelf life of produce for a couple of weeks by getting rid of the microbes that are currently on the surface of it. Of course, as soon as you remove the radiation, any exposed produce or exposed meat will be recolonized and will regrow. But it does allow for a delay. But what I was telling you a little bit earlier is that radiation doesn't necessarily work equally against all sorts of organisms. What they're showing you on the right-hand side is the amount of radiation necessary to kill the organism. So Clostridium botulinum, you need a dose of 3,300 gray, which is kind of a lot. Salmonella is pretty weak. So Salmonella and E. coli, these are pretty common contaminants of both meat and produce, 200 to 300. A lot of the fungi are not very strong either. And then you've got viruses. So both the Coxsackie virus and the foot and mouth virus are related to polio. So they're in the P. coronavirus group. You can see that they can resist a ton of radiation before they would be inactivated. So this Coxsackie virus causes an infection in humans called hand, foot, and mouth disease. It's really common in kids. They get blisters on the bottom of their feet, on their hands, and in their mouth and throat. In cattle, they have foot and mouth disease, which does something very similar, blisters in the mouth and on the feet, and it can kill the cattle. So just because radiation works for some things doesn't mean it's going to work equally for others. So we use it, though, yes?
How much sterilization do you get through microwaving?
Microwaving? I think the reason microwaving would work more is because it brings up the temperature. So it's the heat. So that it's good for like a lot of your foods. But one of the risks with microwaving is with some bacteria, they produce exotoxins, right? And a lot of the exotoxins cause gastrointestinal upset, we'll say. Some of those toxins that are produced are what we call heat stable. So even though you put your food in the microwave, you warmed it up, it's hot. You got it like steaming hot. All the bacteria are dead, but the toxin is not denatured by the heat. So you eat that and you, real quick. So the toxins produced by Staphylococcus are heat stable. And also there's a bacillus species called Bacillus cereus. So putting it in the microwave will kill them because of the heat, but the toxin won't be destroyed. So Bacillus cereus food poisoning is really commonly associated with rice. So the bacteria will grow on the rice, it'll grow in the fridge, it'll produce the toxin, and you're like, oh cool, this Chinese food from last night was so good. And you put it in the microwave, it's so hot, and then 2 hours later you're puking, it's because of the toxin. So that's what microwaving does. It kills them because of the heat, but if there's anything that's not heat labile or heat sensitive, it can still cause a problem. So microwaving is good. I totally recommend microwaving your food, but I don't promise you won't get sick even if you microwave your food. Okay. So we do use radiation, though, in both medical and food applications. It is approved by the World Health Organization in the U.S. for decontaminating foods susceptible to microbial contamination. Here they give you things like hamburger, chicken, spices, can all be irradiated to make them as sterile as possible. Okay. So, whatever you eat is already it's like the Hulk. Right? It's already been irradiated. All right. Other things we can do if we have a heat-sensitive liquid. So I told you about pasteurization, right? Because you don't want to drink orange juice that was boiled. You don't want to drink milk that was boiled or autoclaved. So you can pasteurize it. You can heat it up. But what you have to understand is when you pasteurize something, it still has microbes in it. But there may be situations where you do not want microbes in your liquid. For example, in the lab, if we gave you broth that had some microbes in it already, that wouldn't be very good because it would contaminate your culture. Or if it's some sort of medical liquid that you want to give to a patient, like saline or some sort of fluid, you don't want microbes in it. It has to be sterile, or you'll kill somebody. So what we can do in that case is we can filter sterilize. So it's a physical method. Most organisms, especially if you use a 0.2 micron filter, most cellular organisms will not be able to pass through that filter. Notice I said cellular organisms. Viruses can still get through the filters. Okay. But 0.45 will capture most bacteria, but things like Campylobacter and Helicobacter, which can cause really significant gastrointestinal infections, can pass through a 0.45. So I'd recommend a 0.2 micron filter. So that'll sterilize it and remove all cellular organisms. You may still have viruses in there, but viruses can't replicate without cells, so it's kind of a trade-off. The pores will allow the liquid or gas to get through. So if you have a liquid that for whatever reason you cannot heat up, in a laboratory setting, if we have a growth medium that has a lot of sugar in it, we can't heat it up. What happens when you heat up sugar? It caramelizes, right? And I might like to eat caramel, but my bacteria are not going to do very well on it. So we don't want to caramelize our sugars or any sort of heat-sensitive liquid. We usually will filter sterilize instead. So if it's a small volume, we can use a syringe with a syringe filter. If it's a larger volume, we can use a filter on a flask or a bottle. Okay. And so these are all kind of physical methods. We can heat them up, or we can expose them to radiation, or we can directly filter and pull the microorganisms out. But we can also add chemicals. So we have chemical mechanisms that we can use to control microbial growth. And so when we talk about antimicrobial compounds, there are a couple of different classes. We're either going to inhibit the growth of a microorganism, or we're straight up going to kill it with a chemical. If we kill it, we say that compound is cidal, like bactericidal kills bacteria. Fungicidal kills fungus. Virucidal kills viruses. When we say something is static, like bacteriostatic, fungistatic, virustatic, we mean that it inhibits the growth. So the distinction is important, killing versus inhibiting growth. And you might ask, well, like let's say we're talking about treating a human infection. A lot of antibiotics are actually bacteriostatic, not bactericidal. You're like, well, I have an infection. I want this infection gone. Why won't you give me something that will actually kill my infection? Because for the majority of individuals who have a functioning immune system, as long as you can stop the bacteria from growing, you can stop the infection from getting worse, the patient's immune system can come in and clean it up. So static is good enough. Now, if you have a patient who has no immune system, static is not good enough. You would have to use something that's going to kill to get rid of that infection. So there are reasons why you can prescribe bacteriostatic antibiotics instead of bactericidal. So there's also a third classification they list here called bacteriolytic. That means the bacteria explodes. Not a lot of antibiotics that you would prescribe to a patient are bacteriolytic, but things like detergents are. So anything that disrupts the membrane, if you disrupt a cell's membrane, you usually cause it to burst. So bacteriostatic agents usually inhibit some sort of growth process. They'll inhibit cell wall biosynthesis. They'll inhibit protein synthesis. And so it prevents the cell from growing. Now, eventually, if you prevent the cell from growing for long enough, yeah, it will die. But it's not an immediate issue for the cell. Bactericidal will bind very tightly to their target and will usually kill the cell within a few minutes. And then bacteriolytic mostly interfere with the membrane and cause cell lysis. And so if we were to look at these drugs in a culture, and we were to look at the cell count, and we were to look kind of on a spectrophotometer, each of these things would show us something different. So if we had a bacteriostatic compound, we would see the cell growth. We're plating, we're getting all of our viable cells. And then we add the drug right here. And when we add the drug, growth stops. But we could still plate the cells, and they would grow because they're alive. If you pull them out of here and you try to grow them on a plate, they're alive. But they're not replicating inside the culture. We remove the drug, they grow again. So it didn't kill them, but temporarily we stopped them from growing, which again, for a lot of patients, is all you need. Because you can maintain this for 10 days with antibiotic therapy, and the immune system can come in and start to clear it up. So with bactericidal, what we're seeing in the middle is the total cell count is still the same. Right? The cells are there, but they've died. So when you try to do a plate count, they don't show up anymore. But like when you look on the spec, the level's the same because a dead body is a dead body. And then with bacteriolytic, you would actually see the cells disappear because they explode. But again, most antibiotics that you would prescribe a patient are going to be bacteriostatic or bactericidal. When something disrupts a bacterial cell membrane, it also can disrupt your cell membrane too, because your cell membranes are not all that different from bacteria. So if it causes bacteria to explode, it could potentially cause your cells to do the same. So what I want you to do right now is take a few minutes and think about questions that you would ask before we move on to talk about how we quantify the amount of antibiotics. So what questions do you have about this section? What questions do you think I would ask you about this section? Okay, so take like 2 minutes. So to make sure you yeah. If you're also dealing with like autoclaves because it has the high pressure, it's the steam matches and the energy is a little bit more. But yeah, you're absolutely right. The USP is you want your like 140 or 150 or something like that. So because you're also talking about more dry heat, you have that moist heat and there's no pressure, and so it's a combination of like the way that yeah, that's why. That's a good question. All right, so who has questions for me? Any questions about what we've just covered?
Why don't we use bacteriolytic agents to treat infections?
So bacteriolytic, remember, why would we not want to use a bacteriolytic agent maybe necessarily to treat someone's infection? It could harm all cells. There's another issue we might have to think about. What are our two classifications of bacteria that we talked about mostly? Gram-positive and gram-negative. What happens if I just start blowing up gram-negative cells all over the body?
LPS everywhere.
LPS everywhere, toxin, right? So these are things that are really important to consider when you've got a patient who comes in. If you give them an antibiotic, that could potentially lead to death and lysis of gram-negative cells. You could cause a situation for that patient where they experience the toxic reaction, that cytokine storm, which could be worse than the infection. So as a physician, those are things that you'll have to balance out. Right? I always have the nursing students, why do we have to learn how to do a gram stain? Who cares? And I'm like, well, if you kill a patient because you give them a drug that causes all their bacteria to explode, how are you going to feel? You're like, oh, I feel pretty bad. I'm like, well, see? Okay, so how are y'all going to feel if you're a physician and you just, who cares? I'm just going to give them this one. And it kills them. So you have to think about those types of things. Any other questions that people have or think I might ask?
So, what kind of agent do you use on a patient that has a weak immune system?
What kind of agent do you use on a patient with a weak immune system? Bactericidal. Why?
They need all the help they can get.
because they need all the help they can get so we can kill all the bacteria. So if you have a patient who's immune compromised, you most likely want to be giving them a bactericidal antibiotic. Bacteriostatic usually won't be sufficient because they have no other mechanism to clear the infection. Any other questions? Okay, so when we talk about antibiotics, we also have to talk about this idea that we need to know how much to give. And so in microbiology, the term we most commonly use to talk about this is the minimum inhibitory concentration. And what we mean by the minimum inhibitory concentration, or the MIC, this is the minimum level of drug that is necessary to inhibit the growth of the microorganism. So when you're talking about a patient, it's the level of the drug you need to have in the patient's target tissue in order for that patient to be treated. And we can determine the minimum inhibitory concentration using a couple of different mechanisms. The one that you will do in the lab is called a disc diffusion assay. You take whatever agent it is that you're interested in at a known concentration. You put it on a little filter paper disc. You take a agar plate. You spread a known amount of bacteria on the top of the plate. You put your disc on top of the bacteria, you incubate overnight, and you look for what we call a zone of inhibition. So what should happen is when you put this disc that's full of antimicrobial agent on your plate, that antimicrobial agent will diffuse out of the disc and onto the plate. So it will be encountering the bacteria. If it's able to inhibit bacterial growth, you'll see a nice area around the disc. And that indicates sensitivity. That's what we want to see. If you see your bacteria grow all the way up to your disc, that's a problem because those bacteria are then resistant to that drug. So in the clinical setting, any of you who are considering clinical laboratory science, this is one of the most important things you do. You identify the cause of the infection, and then you determine the susceptibility of the microbe. Because if that's a microbe that's resistant to penicillin and you're giving a patient penicillin for days and days and days, that patient is probably going to die because their infection is resistant. And so this gets really, really dangerous when we talk about infections like tuberculosis, because the bacterium grows so slowly. It can take 3 weeks to grow on a plate. So it could take between 2 and 3 weeks to figure out what drug you can use that's the appropriate drug. So you take a shot in the dark and I'm going to give them this one and hope it works. So your patient can be getting sicker and sicker until you figure out which drug to use. So this is a really important test that you would do in a clinical laboratory setting. Okay. So other types of chemicals that we can use. These are some of these terms I told you that a lot of people use interchangeably, but are not necessarily interchangeable. So sterilants. So something that sterilizes kills everything, including endospores. For the most part, anything that somebody puts into your body should probably be sterile. You want endoscopes to be sterile. You want surgical tools to be sterile. Dental tools to be sterile. Tattoo needles or piercing needles to be sterile. Anything that breaches your body had better be sterile. That's why I say nail salon too. People get some gnarly infections from nail salons if the tools are not sterile. Now, disinfectants are used on surfaces to kill microorganisms, but may not kill endospores. Lysol kills 99.9% of germs. What's that 0.1% it can't kill? I don't want to know. So it's a disinfectant. Hopefully it reduces, in your kitchen, you get all the salmonella off the counter, all the E. coli off the counter, but there might be other things on the counter. Okay. Sanitizers reduce microbial numbers but do not sterilize. Usually sanitizers, we see those in food service. So these are your, dishes and silverware, right? They don't sterilize silverware, they run it through a dishwasher. Warm water, yay. Right? Warm water and some chemicals is not really going to kill much. You've all gotten forks and stuff that already still have like food on them. Right? They still have food on them, they still have other people's mouth germs on them too. I know somebody who actually won't use regular silverware. Like, no, I need the plastic
So
recipes.
jaded to the role now.
You are?
You are.
I am? I don't trust anybody. Everybody's dirty and gross. Yeah. Can I have a to-go cup? Thanks. That's me. And then antiseptics kill or inhibit microbial growth, but are non-toxic enough to be applied to living surfaces. So mouthwash, soaps, okay, Purell. Okay. So things, obviously they're not sterilants, because if they killed all living things, they'd kill your cells too. So they reduce the microbial load as much as possible. And then what we have on this slide here is just different types of antiseptics, sterilants, disinfectants, sanitizers. What you'll see is that sometimes a compound can be used as both. We tend to use alcohol, especially ethanol, in the laboratory setting. It's a general purpose disinfectant to clean off any surface. You've got things like formaldehyde, where if you change the concentration, it goes from a disinfectant to a sterilant. Hydrogen peroxide, ozone. Here's pine sol, a general purpose disinfectant, right? And it tells you the mechanism of action. So lipid solvent, so it helps to break down lipids, disrupts the cytoplasmic membrane. So phenol, triclosan, benzalkonium chloride, which also disrupts cytoplasmic membrane, really common in shampoos. So if you go home and you see benzalkonium chloride, it's a little antimicrobial inside your shampoo. Okay, iodine, all sorts of stuff. So there are lots of different chemicals that we can use to help at least reduce microbial load. And so really at the end of the day, the majority of food products, beverages, toothpaste, all that stuff, it has microbes in it. You just hope it's not too much. And when it's too much, you hope the FDA says, hey, it was too much, and they recall it. But anything that somebody puts inside of your body, like needles, medical devices, you want it to be completely sterile. So there was a big issue at a hospital in Los Angeles a few years ago because they were using endoscopes, and endoscopes you can't autoclave, so they were using some sort of radiation to sterilize them. But they weren't doing it as correctly as they should have been, and so a bunch of patients all got drug-resistant gastrointestinal infections because the endoscope was used on patient A, who had the infection, and then went down the throat of patient B and down the throat of patient C. Okay, so
Did they know?
some of them did, yeah. Some of the people died. So it's really, really critical, if you are going to be a physician, that you sterilize things, or you could, at the end of the day, could kill somebody. Okay, so any questions on the rest of Chapter 5 before we briefly begin to introduce Chapter 6? Yes.